Hello everyone, welcome back to my channel. So today I am going to explain you the transcription in the eukaryotes. This is the full notes of the transcription in eukaryotes. So usually eukaryotes has thousands of genes. Okay, the genes are called the monocystronic genes. In the prokaryotes, which is polycystronic gene. And in the eukaryotes, there are different types of the RNA polymerase, which is RNA polymerase 1, RNA polymerase 2 and RNA polymerase 3. In the prokaryotes, which has only one RNA polymerase. In the case of RNA polymerase 1, it transcribes the ribosomal RNAs. It synthesizes the 45S RNA, which matures into 28S, 18S, 5.8S RNAs. And in the case of the RNA polymerase 2, it transcribes the mRNA and heterogeneous nuclear RNA. That is called the hnRNA. In the RNA polymerase 3, it transcribes the tRNA. 5S RNA and snRNAs. So, usually the RNA polymerase 1 transcribes the rRNA. Means the rRNA 45S, which matures into 28S, 18S and 5.S rRNAs. In the case of the RNA polymerase 2, which transcribes the mRNAs and heterogeneous nuclear RNA, and third one, which is the RNA polymerase 3, transcribes the tRNA, 5S rRNA and snRNAs. Next is the formation of functional RNA. The second complexity present in the eukaryotes, that is primary transcript of RNA format, contains exons and introns. So the second complexity present in eukaryotes, it contains the exons and introns both. So in the case of eukaryotes, splicing occurs. So what is splicing? Splicing is when the RNA formed undergoes splicing, the introns are removed and exons are joined a definite order. The hnRNA, which is formed by the RNA polymerase 2, undergoes two more processes, which is called capping and tailing. Capping means the hnRNA means heterogeneous nuclear RNA is precursor of mRNA, means hnRNA and mRNA produced by RNA polymerase 2 in nucleotide methylguanosine triphosphate, or it can also called methyl GTP or MGPP, is added at the fifth end of hnRNA. So first is RNA being transcribed. RNA with exons and introns. Second step is the capping of mRNA, in which in the fifth end MGPP or methyl guanosine triphosphate added at the fifth end, and introns or exons are shown here. Next is about splicing, means introns are removed and exons are added. Next is tailing. Tailing means at the third end, 200 to 300 adenylate residues are added. This is what capping is. Next is polyadenylation. Next is fully processed messenger mRNA is ready. Means after capping and tailing, the fully processed hnRNA becomes mRNA. This mRNA is then transported out of the nucleus into the cytoplasma for translation. So that's it. There are some easy steps. It has 3 RNA polymerase. It is go to being transcribed. It has both exons and introns. It undergoes the splicing, then capping and tailing. It's a full process. Thank you for watching. Keep supporting and subscribe to my channel.